Hi guys, I'm back. It's Mrs. Horseman, and today we're going to be talking about the second part of Standard 5, which is the replication of DNA. So what's the whole point of replication? Why do we need to make another copy of DNA? I'm so glad you asked. The reason is because DNA has to replicate so that when you have one cell that grows and gets bigger and then divides into two cells, each cell has an exact copy of that DNA. If those cells don't have the exact copy, the exact blueprints or directions for everything that happens in that cell, they're not going to function the right way. And that's when a mutation happens. And if enough mutations happen, that's when you get cancerous cells, and that could be a really bad thing. So good thing for us, our cells do this thing called replication so we can so that our cells can have complete copies of DNA. So when the heck does this happen? Well, there's a whole cycle of how cells are replicated, and we will get into that in the next unit. But for now, what you really need to know is there's a part of that phase called interphase. So from here to here. And that's when the cell does all of its normal cell cycles. It gets bigger, it does stuff like protein synthesis, photosynthesis, cellular respiration, all of those things moving in and out of the cell. That's when all the regular cell functions happen. But that's also when a really important thing happens called DNA replication. So right in this phase, that's pink right here, this S phase, that's when DNA makes a copy of itself, okay? So that way, when the cell, and you can see even here, there's two pieces of DNA here and two pieces of DNA here, so that when that cell gets bigger and bigger, and then it finally divides and it creates two different cells, each cell has an exact copy, an identical copy of that DNA. So how does it replicate? It's a really complicated process. We're just going to go a little bit into the depths of it so that you understand the process of it. You don't need to understand all of the intricate stuff that happens, because I don't even know all of that. But what you do need to know is that when it does replicate, remember we talked about it being complementary strands, so that one strand has ACG and the other strand has TGC. Okay, so it's the complement of it. Well, and then this is what this is showing. Here's the one strand, and then here's the complementary strand, or the matching copy of it, okay? So the very first thing when DNA replicates is that this enzyme, or a special protein called helicase, comes in and it acts like a zipper, okay? It's going to uncoil or straighten out the DNA. So DNA is all twisted up. It's going to straighten it out so that it's like this, and then it's going to come in right here, and this helicase is going to open up that DNA strand, okay? And it does that by breaking these hydrogen bonds that are between every single matching base pair, okay? So that's the first enzyme, is helicase. The second enzyme is DNA polymerase, and that is signified by this circle right here. There's one here and then there's one down here. So each polymerase is going to come in and it's going to move along those open bases, and it's going to add a new nucleotide to every single one of those bases, following the base pair rule, okay? So it's going to make the complementary base pair on the new strand of DNA. So what is the final product? We are going to end up with two identical copies of the original DNA molecule. And if you see here, our original strand is blue. Our new strand, we have blue, which was the original strand, and then we have a copy, the matching complementary strand, of yellow. And then again on this side, blue, and then a complementary matching strand of yellow. Okay, so here's exactly how it works. Like I said, helicase is right here, and it's going to come in and unzip that DNA strand, all right? So it's going to break those hydrogen bonds and make that DNA pull apart. And then the DNA polymerase, which is that circular structure, is going to come in and it's going to add new nucleotides back on, and it's also going to proofread it. So it's going to make sure that it's doing the right job, because guess what? If it adds the wrong base pair, the wrong nucleotide, that's a mutation, and that's a bad thing. So your cells do as much as they possibly can to make sure that those mutations, that those wrong pairings aren't happening. All right, so I will show you this replication video while we're in class, but there is a video that kind of shows the whole process going along. All right, so I just want to check with you really quick. Take a few seconds and write these answers down in your notes, and then we'll definitely talk about them in class. So the first question is, what is the whole point of DNA replication? We talked about this in the video, so kind of think back and look at your notes and see what you wrote down. All right, next question. What is the enzyme that unzips, that acts as a zipper and opens up that DNA strand? Question number 3. What does DNA polymerase do? Got that? All right, number 4. How many copies of the DNA are made at the end of the replication process? All right, we're almost done. Number 5, why is DNA unique to an organism? And the very last question, when is DNA copied? So what part of that cell cycle is DNA copied? Why does that happen? All right, I hope you got those answers. If you didn't, you need to check back in your notes or even rewind this video and watch it again. And we will definitely make sure that we talk about those in class.